What actually is a stem cell? Well, stem cells are cells that do not have a set function. As a result, stem cells are able to assume the role of many different cell types during early stages of growth. Stem cells also possess the remarkable power to divide without limitation and assume the role of a variety of cell types as long as the organism is living. This, as you can see, could be very beneficial for medicine.
In order to obtain large amounts of stem cells, scientists will undergo a cloning process using an initial stem cell. This process of cloning is called somatic cell nuclear transfer. Somatic cell nuclear transfer relies on the fact that traits and the future function of a somatic, or body cell, are all programmed in the DNA of a cell. What makes stem cells so special is that they're not special, or rather specialized. A specialized somatic cell is one that performs a specific function in the organism. For example, in a human, skin cells or organ cells are all specialized cells. As a matter of fact, the majority of somatic cells in a human are specialized. But if all the somatic cells in an organism contain the same DNA, how do stem cells not perform the same functions as specialized cells? Well, it all starts with the structure of DNA in a process called transcription. A DNA molecule consists of a sugar phosphate backbone that is arranged in a double helix structure with 4 bases in between the backbones to serve the role of data storage, just like binary in a line of code. Transcription is the process of breaking apart the helix and separating the bases so that we can copy them in a molecule called mRNA, which allows us to synthesize proteins. This leads us to the answer of the question. The reason that stem cells and other somatic cells are different is that epigenetic tags inhibit some genes from undergoing the process of transcription, preventing the synthesis of mRNA, meaning that the proteins a certain gene is responsible for translating will not be produced, rendering that gene ineffective. In a stem cell, these epigenetic tags essentially block off certain regions of genes, causing the cell to not have a specific function. This is why stem cells are so hard to produce. The epigenetic tags get removed as the organism develops, specializing the cell for forever. Now that we know that stem cells are hard to produce, this takes us back to the somatic cell nuclear transfer. This is a method of cloning where the nucleus of the stem cell is removed and placed into an oocyte, or female egg, that has already had the nucleus removed. From there, the nucleus takes over the cell, turning it into a stem cell, and then going through the process of mitosis.
There is a significant amount of controversy revolving around stem cell cloning. Much of the controversy comes from ethical concerns rooted in modern society and politics. On one side of the debate, there are people that believe stem cell cloning is beneficial for medicine but should not be used if it interferes with an unborn child's right to live. On the other side of the debate, there are many people in the fields of science and medicine, or simply patients, that think it would be impractical to not use stem cell cloning if it can potentially cure diseases and heal injuries. Stemming from this debate, no pun intended, comes the political controversy in the U.S. of whether to use government funds to sponsor stem cell research, how to regulate stem cell cloning, and how the government's decision will affect the future of genetics. Proponents of government involvement believe stem cells can open new gateways in science and DNA technology that will greatly benefit the medical and scientific field. For example, cloned stem cells can be used to grow back damaged tissues, cells, and body parts in patients suffering from burns, diseases, and other ailments or illnesses. Opponents of the government providing funding to stem cell research believe that taking stem cells from an embryo is immoral and taxpayers' dollars should not be used in such fields that interfere with human rights, especially when the status of an embryo and if it is human or not is unclear. The drawbacks to cloning embryonic stem cells are that it is still a fairly new technique that has not yet been perfected, and that despite extensive research, there is still uncertainty revolving around its use in humans. But just like any cutting-edge method, time will tell if its drawbacks outweigh its successes. As of right now, scientists are fairly certain the successes of stem cell cloning will only increase as the process becomes more practiced and studied. According to Shukarat Matilapov, a professor at Oregon Health and Science University, He estimates about 50% of the success can be attributed to the quality of the eggs, while the remaining 50% is related to the optimization of the process. So far, the technique appears to be pretty efficient. From 8 eggs, the group generated 4 embryonic stem cell lines. In the future, Matilipov anticipates it will be possible to produce a stem cell line from each donated